Welcome back after the coffee break. So we'll start with any pending questions for Rakesh on his talk, and then go on to a more generic discussion. Some of the points Rakesh and I have shortlisted and written down, and then I have various different Brownian motion trajectories I can take based on where the discussion leads to. Can you all hear me? Any pending questions? Yes, please.
I have two things to ask. So this is basically general concept. So we in mammalian system are taking a particular species or organism. So DNA sequence is completely same for all the cells, irrespective of which organ it is. But you had mentioned that in maybe liver cell, the expression of the genes differ in that particular genome, the cell, compared to the brain cells, neuron cells. So which means the chromatin architecture or the subnuclear compartmentalization is different between different organs in the cell. Is it like a customized for each of the cell type?
Yeah, so, absolutely. I mean, I'm so happy that you got it exactly correct. Yeah, that's how it is.
So if that is the case, can we not come up with organ-specific chromatin architectures by some techniques? Just okay, this so cell type, this is the architecture we observe.
No, you're very right, and that's what people are doing.
Okay.
Now I talked about mod and code and encode. There's two databases. Their purpose is this kind of thing only. Take liver cell and do all kinds of high throughput, including accessibility and all, and put in the database. So one of the problem with this system is the heterogeneity of cells. Even a liver will have many different kinds of cells. So most of the modern code and encode database is on cell lines because they are pure one kind of cell. So as you rightly said that each cell type will have its own detailed organization. So now if you are dealing with 2-3 cell types and these are all high throughput methods, you will see the average of that. Yes. So that is the big constraint. But having said that, now there are technologies, people are claiming that they can do these things at a single cell level.
Okay.
So for single cell architecture, if you can define, that will be the ultimate. There are certain difficulties in doing that, but it's probably going to be possible. So if single cell level you can do, then you can very precisely tell this is the whatever feature you are talking about, packaging or particular modification or expression status, these things. Then we will be able to figure out.
Okay, so that's also by tracing a one cell.
Yeah.
So we can also study the chromatin architecture changes through the developmental stage of the cells as well.
Yeah. So
which also varies as the development.
So like
differentiation happens.
Yeah. So we talked about differentiated cells. Now there are intermediate stages
of the
and what happens during that? Yes. How many steps are required for a let's say totipotent stem cell to lead to a final liver cell, for instance. That will be a fantastic thing to see because this is so complex that you cannot suddenly convert a liver stem cell in one step into a particular cell type. It has to go through many stepwise changes. Then only a final product comes that precisely the genes that are required for a particular cell type are now accessible and those are not required are now buried in the inaccessible part of the genome. So this is going to be a multi-step process.
Yes, sir. The second thing is about the conservation of non-coding sequences. So apart from the repeats, so the genome made up of many large portion of non-coding regions, and basically the coding region is defined by the length of the ORFs. Mm-hmm. So it basically 200 or 300 length, base pair length. If the non-coding region have the shorter ORFs, and there has been many reports because of the ribosomal profiling, they have found that the short ORFs indeed give functional peptides which regulate the proteins as well as the gene coding counterparts. So the conservation of the non-coding regions is probably due to the presence of small ORFs.
Okay, so it's good point, but that is actually not the case. So for first part, what you said is right. People have missed a large number of short proteins, very short proteins, because the computers tend to ignore that. But people have now started doing experimentally, because proteomics is very powerful now, and even transcriptomics. Many of the annotations are going in reverse order, that it is there, go and map it. Right? So people are doing that. But having said that, once you know this particular region, then you can tell for certainty whether there is ORF or functional ORF or not. Those things can be figured out. And there are many other things associated. ORF is not enough. There has to be translation initiation side, there has to be stop side. There are many other features that you can tell where there will be a protein or small protein formed from this region or not. It's possible to figure out. Yeah.
Okay. Thank you.
Yeah.
Can I speak up? So I can be heard. My point is just a comment on what Kalini said just now, and something else I wanted to add to that. So the comment is, if it's a short ORF, it doesn't matter, it's still an ORF. Therefore, you're dealing with a coding portion of the genome in a formal sense. So this doesn't come under non-coding. And you can think of the zillion and one possible reasons why that coding sequence is maintained. So this doesn't come under non-coding. If it indeed is an ORF, however short the polypeptide that's coded by it. What was the second comment now that I had? It will come back in a while. Yeah.
What is the ORF?
ORF is an open reading frame. It's a contiguous sequence of nucleotides in DNA, which can be functionally read by RNA polymerase eventually to give rise to an RNA sequence which is translated. That's the important point. So you don't end the story with the RNA sequence. That's ORF, open reading frame.
Technically, it can be changed into a peptide sequence.
True. But this is a trap that we have set for ourselves. This is like this famous 5% criterion for deciding something is significant. So if your p-value is greater than 5%, you throw it out of the window. But that's because you have decided it. There's nothing more significant in that. But now I remember what the other comment that I wanted to make, which is we should keep in mind, and I think Rakesh brought this out very well, that DNA wears many hats. One of them is in terms of the genetic code. And I think, strictly speaking, I was going to bring this up at 1:45, but I'll mention it now since it's come up. Strictly speaking, I think we should restrict the phrase information content of DNA only to that part of DNA that carries encoded information. So information in the linguistic sense, if you wish. So you can think of that part of DNA as being symbolic, with the meaning in the symbol being eventually the protein. But there's a great deal else that DNA does, and which was brought out really nicely in the previous talk, which is essentially chemistry, which we are beginning to understand bit by bit. Probably quite a bit of it physics, which we are sort of groping our way towards understanding. And finally, there may still be a substantial amount left over, which in a functional sense is meaningless, which is really baggage that has accumulated. There's a comment that Kaustu made earlier about repeated DNA sequences. The best bet we have as of this day, and like all bets, this could be wrong, the best bet we have as of this day is that these are relics, remnants of parasitic DNA sequences which expanded, and then through mechanisms that we understand reasonably well, reached a balance via natural selection with the fitness of the organism. But they are there now, like it or not. So they are poised in this pseudo-equilibrium state, if you wish. So you can either get rid of them, in some cases you can take advantage of them, which has happened. And there, of course, you have to be careful when you see them playing a functional role. you should be careful not to interpret that as being the reason why they came there in the first place. This could be overlays on historical processes which you can only guess at. That's all I have to say.
Hello. Yeah. So I had a couple of questions. For this Mars, when you say matrix associated regions, what is that matrix? It's not the lamin, right? It's something within the nucleoplasm, right? So what are the constituents of that matrix?
Matrix is bio. So biochemically, matrix is you take the nucleus and extract out what is diffusible under high salt conditions. Yeah. So you take out histones, you treat with DNA once you take out all the DNA which is accessible, and then you are left with something which is non-diffusible component. It has about 7-8% of the proteins, nuclear proteins, about 20% of the nuclear RNA, and maybe 2-3% of the DNA, which is not extractable even at 2 molar salt. So that's what we call it a nuclear matrix, and there are some other criteria to it. A certain kind of 10 nanometer fibers are the unit of this structure that is formed typically.
Yeah, but this is not the lamin nucleoskeleton, right?
Lamin is part of it.
Yeah.
So lamin in the beginning will show only at the envelope if you see in the nucleus as such. But if you do matrix preparation, extract out histone, then you can see meshwork of lamin even more interior, which means antibody is not able to reach the lamin fibers or lamin protein in the more interior of the nucleus.
Those are, Drosophila doesn't have A-type lamins though, right?
Yeah.
So it's just B-type lamins inside the
you can see nice circle. Yeah. And so there are hundreds of proteins actually, 700-800 of protein in the matrix. And there are some RNAs very important. If you treat with RNAs A, the matrix will collapse. So that means it plays a structural role. Actually, one of the RNAs we identified and we kind of knocked down in the flies, and they give weird phenotype. Nucleolase is fragmented, heterochromatin is fragmented. So that means architecture is somehow affected. And these larvae die at pupation stage. They never pupate. So they pull up to that long, but then they die. And the proteins are, as I said, few hundred proteins, because we have done lots of work on this. So if you take different cell types and you make matrix from them and compare, so only 40% of the proteins are common in all of them. So we call them core numat protein. Remaining ones are different. So there is different cell type is specific, nuclear architecture component is variable.
Yeah. Yeah. No, this will be important because that's where your loops, if they are there, are anchored, right? And the other thing I was wondering, this ACC, TCC, which is simian-specific repeats. So is it essential to making the simian, or is it incidental, that it arose in that ancestor, as you said, and then it has been retained?
Everything is accidental, that is, there's no, this doesn't come by any design. It's just that now it becomes useful. I don't know whether it is essential for simian, but now there are ways of doing it, and we will figure it out because they are intronic largely. And we are guessing that they must be playing role in splicing of those proteins where they are intronic. So in simian and non-simian, if the splicing is different, then we can, at least in cell types, we can go and remove and show that splicing becomes different. So when you narrow down to something specific, you can now do experiment. That's what genome editing has enabled us. Now we are kind of getting free of the model systems. We can actually change any cell type as long as we can grow the cell of the organism. So it's possible to, but we can't say whether it's essential. It's just that we see it maintained. And like Vidya said, that everything that is maintained is maybe by chance. Some of them may not lead to anything in our functional assays.
Yeah, I have a very naive question. Are these epigenetic modifications seen only in eukaryotic cells, or do bacteria also show variations in gene expression profile with the same genetic?
They don't have these kind of, first of all, bacteria, most of the epigenetic modification, when people say it's either histone tail modifications or sometimes DNA modifications. And since bacteria doesn't have histone and other thing is bacteria, except a few genes, most of genes are required in one cell cycle, most of the genome plays out. While in higher eukaryotes, most of the time you don't require most of the genes, they have to be silent. So a lot of negative regulation has to be there. So you need more robust mechanisms. While in bacteria, very few need to be repressed. So by a repressor you can manage. So there's large difference. So very little, I'll say, epigenetic mechanism that are in bacteria. But we should remember that DNA methylation, all these are used by bacteria to protect themselves from phages. The restriction system has emerged from there. It's not working.
Few papers by ENCODE have indicated that the level of transcription actually is much more. I don't think they've done experiments at the whole genome level, but several megabases indicating that close to 90% of the DNA is actually transcribed. I haven't seen follow-up of those papers functionally, or many people indicating of what value that is. Is that a sort of housekeeping warm-up mechanism or what it is? Do you have any comment on that?
Yeah, this is actually that most of genome is transcribed is not new. Now we have new methods of verifying that. In early 90s, the paper that most of the genome is transcribed, there's PNAS paper, it was very surprising that if you look. So, but quantity of the transcript and its stability and all those things need to be whether this is functionally relevant or like you said, it's some kind of pre-run or warm-up, like you said, is not known. But this is that's why this RNA field is becoming very, very exciting because in many times now they are turning out to be very, very important components in regulation, but very little known.
Came across a quote relevant to this. I just quickly read. It's okay to be white. It's okay to be black. It's okay to be Asian, it's okay to be Hispanic. It's okay to be female, it's okay to be male. It's okay to be gay. It's okay to be straight, it's okay to be you. But it's never okay to underestimate the importance of non-coding DNA.
Okay, I was trying to convey the same in hours of...
Suren, sorry to strike a sour note to this very nice. Well, anyway, as far as I know, it's your quote, so but sorry to strike a sour note. I think what is okay, and we don't have enough of it, is to be skeptical about claims as to functional roles for anything in biology. There's a huge amount of noise floating around. And actually, it's a point of psychological interest why people are uncomfortable with noise in biology. But it's there, it's been well established, and the possibility that it's more prevalent than we suspect should not be ruled out.
Absolutely, yeah.
Maybe I should tell something which is different from what we have been talking. You know, Sidney Brenner once gave a talk in NIAS. This links to the quote that you read. And there was talk on junk DNA and all. He sort of brought it up, trash we keep, junk we throw and all. Many of you may have read it. So one among the audience asked Sidney Brenner, and most of you are perhaps familiar with his one-liners. So what do you think of this junk DNA? Does it have a functional role or not? And he asked the gentleman, what's your age? And the gentleman replied back, he said, I'm 58. He said, you don't worry about it. Leave it to the guys who are 38. Were you there in that?
Sidney Brenner, I think we can ignore some of this.
So I have some comments. I think whether a repetitive sequences would be present in a genome or not depends on depends on many other factors, and one of the factors is how the repetitive regions are suppressed, how they are not allowed to being expressed in terms of at the level of transcription. So we know that we have worked on an organism where RNA interference is present. So all the genes of RNA interference, Argonaute, dicer, and RNA-dependent RNA polymerase, these genes are present, and RNA interference is a mechanism of suppression of gene expression. So, and we looked at a species which is very closely related to that RNAi proficient species. And RNAi proficient species has a lot of repetitive regions in the genome, but RNAi deficient species lost most of those repeats, including some of those active transposable elements. So I think one has to also look at not only the presence of repeats, but presence of mechanisms to suppress the expression of those repeats, if they are actually transcriptionally proficient or not. So to test whether RNAi is really suppressing the transcription of repeats and further rearrangements in the genome, we deleted RNAi genes in RNAi proficient organism and grew them in the lab for 1,000 generations. We just kept on subculturing them to see whether RNAi is really responsible for keeping those repeats intact. And after about 1,000 generations, when we analyzed the genome, we found that there are certain repeats which are lost. So I think it's a coexistence of the suppression mechanism and the presence of repeat. Both should be considered at the same time. The other comment that I want to make is, why do we have like repeats present on different chromosomes? Whether that has any other relevance, and you alluded to that sometimes those repetitive regions cluster together in 3 dimensions. So again, with the simpler organisms like the various yeast species, we looked at that whether the rearrangements you see in the genome or the chromosomes, whether they involve those repetitive regions or not. And we see that these similar or almost identical regions from different chromosomes cluster together and are involved in interchromosomal rearrangements, probably through homologous recombination. So, and that's one of the ways by which you can get evolution of species, because many of the organisms are asexually propagated. They don't have meiosis to generate variations in the genome and the gene expression. And the last comment that I have is whether there is epigenetics or epigenetic regulation in bacteria. I think David Ellis wrote a book where he explained that what he meant by epigenetics. And one of the classic examples that he has given is it's like a switch on or off. These two states can coexist, and whether it's on or off cannot be really predicted from the genome sequence. For example, if you have a Lambda DNA, which is a virus for bacteria, if it infects a bacteria, whether it will be integrated into the genome of the bacteria, which is called the lysogenic cycle, or whether it will kill the bacteria, which is called the lytic cycle, it's unpredictable. But it can take one of the two modes, which is like whether a switch is on or off. And what he referred to is that this kind of mechanisms where a gene can be turned on or off, or the fate can be one way or the other, is probably the epigenetic mechanism. So I think bacteria also has an epigenetic regulation of gene expression or fate determination.
Yeah, I think what you say is perfectly fine. And this is something which I was trying to show that many things you mentioned is true for simpler organisms. There's lots of fluidity, there's a lot of dynamics linked to RNAi machinery and so on. So those are the guys who are more flexible, and more complex organisms are becoming kind of inflexible in these mechanisms. So maybe after some time, before we destroy this planet by our activities, maybe we not survive evolution beyond. Those who are more flexible, they will survive because they can adjust to many things. There's more fluidity in their system is still remaining.
On all those points of, again, how exactly to define a complex organism, how complexity emerges, points that Madan raised. So we'll come back to that in a moment. So since the aim of the meetings, as I talked to the organizers, is to generate more dialogue between people coming from different ends of the spectrum. So I would like to take the question, what exactly the word complexity means, but that question is too complex for now. So we'll start with a simpler question. So since many of the words uttered here, written here, were talked about by various peoples, both today as well as yesterday, going back to Kaustubh's talk on epigenetics. So I would like to start with just a general show of hands by what exactly do we understand by the word stochastic. You talked about epigenetic, lambda, on, off, all of those aspects. So let's just start with because here I think we'll see a clear divergence, two clusters being formed, what biologists understand by the word stochastic and what physicists understand by the word stochastics. I'm trying to see if that actually happens. That's my hypothesis, and I'll be happy to be proven wrong. So just a quick show of hands. How many of you, when you say the word, read the word stochastic, think that means the mechanism is unknown? Can you please? Okay, so the all options are mechanism unknown, unpredictable, random. Some of you may think these are synonymous, but not necessarily. A combination of the above. You can make a weightage factor of this, 0.5 times A plus 0.2 times B plus 0.3 times, or any nonlinear combination also, or none of the above. So D, okay, so let's start with A. How many A? Mechanism unknown. If you don't know the mechanism, it's stochastic. I've heard this many times. I've seen talks, stochasticity in signaling, and there is no distribution. Again, if I see a title, stochasticity, I expect to see some distribution, some flipping, but there was none of those graphs in those talks.
I think it's context dependent.
Oh, that's another word I just love, fortunately or unfortunately. All right, so mechanism unknown, do you think that is stochastic? Okay, unpredictable. Okay, so unpredictable, 2, 1. Random, wow, I'll just say n much greater than 1. A combination of the above, so n greater than 1, n approximately equal to 1. Okay. None of the above. So, okay, so at least there is some convergence that it's, one of these means something in that context. Now again, I'll connect this to the idea of genetic and epigenetic in a moment, because genetic, as Kaustubh's initial slide yesterday talked about, genetic, again, the Mendel's laws, and there is, if you know the genotype, you know the phenotype. It's, again, in physics terminology or in modeling terminology, it's a very deterministic system. If you know the initial condition, you know the final condition, what the system will reach to after a given point of time. That is what Mendel's laws are, right? That you can actually calculate the frequency of a particular allele or frequency of a particular gene, knowing how this happens. Now, this is the word which many of us have a hard time wrapping around, that what exactly is non-genetic, what exactly is epigenetic, what is somewhere in between, and are genetic and epigenetic really that black and white, that if a mechanism is being regulated genetically, does it necessarily mean that there is no non-genetic component to it? Or if we don't know the genetics, again, does it always have to be non-genetic? So just some discussion on that, and again, going back to Kaustubh's point in that context, that the switch can be on or off. So even if it is the same genotype, you can actually have multiple outputs, which is what sort of leads to these ideas of stochasticity, that it is, you have a given condition, and from that given condition you can actually have multiple outputs, such as the coin flip. So it is unpredictable, it is random, yes, it has all these features, but mechanism is known in some ways, right? You know how to flip a coin. So mechanism unknown is not necessarily the most accurate description of the word stochastic, but yeah, all of these features do tend to be. So just, again, starting this discussion on that end. So again, how many of you think that genetic and non-genetic, or again, just as we can clearly say coding and non-coding, coding is something which will actually be translated to a protein, then it may or may not have a function depending on the conformation of the protein, the structure of the protein, and so on and so forth. So the function is a whole new level. But at least we know what is coding and non-coding. Do you really think we know what is genetic and epigenetic, or can we really segregate that as clearly as possible, or do you think that there is some crosstalk, that there might be some non-genetic component which might get stabilized later or something? So any thoughts on that idea in general, again, in the context of what Rakesh was talking about, the evolution of that or otherwise? Yes. So how would you define genetic contribution? Coding is clear. That's something which gets converted. So do we think that genetic is equal to coding, or is there more than that?
Can I comment on something you said before Rahul's question? Which is, again, we should keep in mind that these are words which have been around a long time.
Yes.
And they have got a sort of entrenched meaning attached to them. And we should be a bit cautious in trying to come up with meanings of our own. So just 2 or 3 comments. One should distinguish between genetics and genesis, and the prefix epi is attached to both epigenetic and epigenesis. Genesis, of course, goes back to the Bible at least. Epigenesis goes back to Aristotle in the context of biology, where he described development as an unfolding of latent possibilities. And that's roughly the sense in which epigenetics is also used today, as an unfolding,
yes,
figuratively, unfolding of the information latent in the genome. So these are the senses of the words which we have today. I think Kaustubh, I'm not sure, referred to Waddington as having coined epigenetic. Actually, it was Sewell Wright some years before Waddington, but it's Waddington's name that's alluded to very often. Nonetheless, the idea here is of something hidden, at least cryptic, which is being made manifest through a process or series of processes, and they are referred to as epigenetic processes. A genetic change conventionally is meant a change which can be seen in the primary DNA sequence of the organism. Now, if there is a change of whatever type and at whatever level you're looking, which cannot be traced back to a change in the primary DNA sequence, it has become conventional now, and that's a neologism. We have created the word. It's become conventional to call that epigenetic.
Yeah, and then again, going back to when this term epigenetic was claimed, then all of the molecular mechanisms which we know today were not, of course, known at that point. So now we are beginning to elucidate some of those mechanisms, as Kaustubh alluded to chromatin modification, and also Rakesh talked about some of the other aspects of the entire non-coding DNA which might be contributing to that. So yes, your point that we should be very careful in having these words, and that's why whether it's really I mean, is there other because in addition to chromatin changes, there are other aspects also which can contribute to, which can still not be traced to primary DNA. Such, again, as you pointed out, the role of noise in the system in that aspect. If there is a multi-stable system, then it can go back and forth in multiple cases. You don't necessarily need a chromatin modification to actually maintain that setup. The chromatin modifications might help increase the frequency of transitions one way or the other in that. Yeah. So yes, and then so that was sort of the general big question that we all, Rakesh and I thought of, and the other was going back to some more specifics, which is starting with either simple sequence repeats or any other non-coding DNA components that we talked about, were the questions on evolution as to can we actually talk about a mathematical framework or computational framework or some ideas from the theory concepts where we can say try to identify, is there a specific trend that we see in the percentage of transposable elements across different species? So if you map it onto the speciation tree, you can actually see here, because Rakesh multiple times referred to what percentage of genome did each of the specific cases, the simple sequence repeats or the matrix associated repeats, how many of them attributed to those components. So that was one thing. And the other, again, going back to the central idea of what exactly is the function of these, because if these are non-coding and they are not junk, they have to play, or they can play some function. One of the examples which he demonstrated was in the context of enhancer blocker, where once you insert this simple sequence repeat, then the enhancer was no longer able to talk to the promoter, and therefore the transcription of the gene went down in that context. So those are the two, if Rakesh, would you like to add any more main questions here? But those are the two main questions we thought we'll at least start the discussion with. Can we come up with some aspects of these other functions, like how exactly physicists understand the term function? Can we look at a theoretical framework to try to address what is the function of these simple sequence repeats vis-a-vis the function of the coding DNA component of that? So I have two relatively recent papers that I can pull up, each taking a mathematical approach to address these questions. But before we go into specifics, I'll be happy to take any other comments or questions that you have, or any thoughts based on what Rakesh said or based on this general picture.
This has to do with levels of organization. We've become fixated to thinking of the DNA level as the only level that's meaningful for understanding how organisms function or systems function. But there are quite obviously other levels, the level of the cell, the level of the tissue, the level of individuals themselves, and much more so the level of ecosystems. And none of them, in the simplistic sense of the term as it's being used here, have genetic explanations.
So I just want to make one comment. So, which probably can explain that what is genetically regulated and what is epigenetically regulated. So let's consider a piece of DNA where a protein can bind and perform its function. Now let's say that piece of DNA is 100 base pair long. And if you have that piece of DNA, then a protein can comfortably bind there and perform its function. Now, if you make it shorter and shorter and ask the question that whether the protein can bind and perform its function or not, what you would see that when it's really shorter than a critical length, then maybe 1% of the time or 10% of the time, the protein can still bind and perform its function. Now, so when you have this 100 base pair sequence and the protein is binding there, it's probably a genetic phenomena. But then in this 10% of the cases it's binding, it's probably, it takes a conformation and the protein comes in proximity, and that's how the protein can bind in the 10% of the cases, is probably not a genetic phenomena and can be a genetic phenomena but at that particular cell cycle. But what is quite amazing in biology is you see that that 10% of the population remember that, the protein was bound to that truncated DNA, and, in every generation for many generations, they can perform that function. How that is, getting inherited to the next generation is probably not understood, and an example of epigenetics. 100% cases it worked.
Yes, I mean, again, like how much of it is actually transmitted to generations and to what number of generations? Because genetic term, at least the way I understand it, and please correct me wherever I'm wrong, it extends for a relatively large number of generations, right? And the epigenetic may have some memory, as Rakesh alluded to and as you alluded to, but that memory may have a faster decay time. That it may have a faster decay time. So to study that, perhaps we need a more sort of long generation studies in that aspect. And as has been alluded to, this is just one example of a review recently written in the context of drug resistance in cancer, but also it has been seen in drug resistance in various antibiotic resistances, that it's not really just one or the other. So what these experiments, or generally speaking, these experiments talked about is that if you take a given antibiotic treatment as just the concentration or in terms of time, and the y-axis is the percentage of cells alive. So what was observed in most of these experiments was that a large number of cells quickly died, but there was a small number of cells which were left behind. Now, this is not surprising, but what the most surprising thing to them was that if you take these cells out, reculture them, and regrow them, you again see the same trend, and you continue to see the same trend over multiple generations. So if this was completely due to genetics, which would lead to a sort of irreversible case, then this graph should look more or less like a flat line. But then later out, at least in this particular context, they figured out that this was due to chromatin changes and established there was an underlying bistable system which was determining the relative frequency of these two states. And that's why cells were actually able to go back and forth in this case. But again, even for a larger number of times when this experiment was done, they actually found new genomic mutations, which were not seen earlier. So perhaps it's an issue of, there is some threshold, there's some tipping effect after which these epigenetic solutions, which the cells have figured out to adapt to various conditions, actually get logged in some way or the other. It can be a mutation, it can be any of the other aspects that Rakesh spoke about.
So can I ask this curiosity? So in this experiment, was it, if you start with certain number of cells which are sensitive to the drug,
yes,
the emergence of mutations was more frequent in the cells which have been harvested later and have epigenetically gone into resistance? Was the mutation frequency more in those, or it is the same, whether they are trained or untrained?
I don't think they did that experiment. They just cultured it over multiple generations, and then in bacterial literature, it's called persisters. So they identified these persisters because they are not resistant. So they didn't use the word resistors, they used the word persisters because they were just persisting it. And when the drug was removed, they actually started proliferating again. So it was again a reversible change, a non-genetic change in that context. But later on, some of them were actually, not all, but some of them were actually shown to have locked those solutions. And now if they were exposed to a different drug, they were sensitive. Because again, they have locked their solution, so they have lost the plasticity. So it is a trade-off to some extent, that how much plasticity a particular cell has, how much adaptability that particular system has, vis-a-vis the ability to lock their solutions in some ways. So now coming to more specific things which Rakesh talked about. So this is just one example, and I'll briefly walk through for just quick comments. Here they were trying to actually model this dynamics of transposable elements, which Rakesh talked about. They did this in both continuous as well as discrete setups, and I'll go to the equations in a real quick. But the general idea was that there is some data available in terms of the frequency of both sort of partial length transposable elements as well as full length transposable elements. And they were looking at, because again, as Rakesh mentioned, that in the context of simple sequence repeat, the mutation rate increases in many cases. So if the mutation rate increases and then there is competition that is also going on, so how can we look at those two interactions and try to get to some steady state behavior which we can then map onto various species and figure out species specific parameters which could explain this particular dynamics that has been observed. So the general setup that they had was, again, these transposable elements, which are these self-replicating DNA sequences. You can actually get more copies of transposable elements. It's more like a copy and paste mechanism. These are first converted to RNA based, and after which, again, you get an identical DNA sequence, which is then later inserted into the genome in this context. So the mathematical framework that was used here was that, so they actually talked about multiple different events, one of which can be deletion in the genome. We have been talking about insertion, but as Costa was alluded to, there can be repression of these, or there can even be deletion of some of the parts of these cases. So they talked about deletions, they talked about insertions in this context, and they also talked about the replications of the transposable elements. Those are the three main mechanisms they talked about. Now, in the context of deletion, they mentioned that most genomic deletions actually are not more than 5 base pairs in length. So then they had this particular set which would talk about all different sets of mutations that you can get. That set is denoted by D. For instance, the deletions of length 1 base pair or 3 base pair, that is the set shown here. There's a given cardinality of the set, which means how many number of elements are given in that set. So in this particular case, there'll be only a 1 base pair deletion or a 3 base pair deletion. So C is equal to 2 in this case. Then, now let's say if you have a transposable element of length I and something of length F can be deleted from this. So how many combinations are possible? So that is I minus F because plus 1, because you can have a deletion beginning right here, you can have a deletion beginning later and all the way up to the end. So they talked about these different, how many different number of deletions are possible, because this is a linear sequence, so it's only one context here. And then that can be divided by the total number of genome size T. So you can calculate the probability that a deletion will happen. Similarly, you can calculate the probability that an insertion will happen, which is being calculated here. They are talking about a more generic case. Yes, I don't know if it is possible to control where the deletions happen. They are talking about all different combinations and then trying to fit the parameters and then see if they can actually get a reasonable fit or not. So I guess that is one of the inherent hypotheses that they're checking based on this model by assuming all of these combinations. So this is sort of full deletion where the entire entire element of length F is deleted. Then they also have partial deletions, and then they have the probability of that deletion happening through that partial deletion as well. So instead of I, you now have this component J here, which can be then can be summed up. And then they have this rate of insertion, which can result in loss of the existing length I, because once you have an insertion, then the length changes. And since the length changes, you have a deletion in the component where the length was I, and an addition in the component where it's I plus 1 or I plus 2 or I plus F. So then they talked about that math here. And finally, they talked about this replications in this. So they did this in both discrete as well as continuous setups. So in the discrete setups, they're basically tracking the number of partial length transposable elements of length I as a function of time. And finally, they look at some steady state behavior. Again, an underlying assumption being that evolution has happened to a large extent. So even if you get the data a few years down the line, that time period is going to be much less as compared to what the evolutionary time scale is. So you can approximate this as a steady state behavior response in this case. And then they also have a continuous dynamics version of that. So I'll go to the results. Yes. Oh, Mike is okay.
So at the abstract level, what you're trying to do is just there is some string and you're trying to... Can you just tell us what can be measured and what are you trying to model?
Yes.
It's a very abstract
thing. It's a
thing.
very abstract model. So what they're trying to measure...
No, experimentally what can be measured?
Experimentally, the frequency of these transposable elements can be measured. of different length, because that's what Rakesh was talking about.
Which is basically the
Monomer,
SSR sequences that...
monomer, dimer, tetramer, all of those combinations.
Okay.
And if you have a given DNA of a given species, then you can model what the frequency of those different monomers, dimers.
Sure. And the addition, deletion, and the insertion, all these things are happening at a generational level, or when? What is the time scale? I don't understand.
The time scale here is again very abstract. There is no generation per se. There is no cell division. It's just at every time unit there is a probability of insertion, there is a probability of deletion, there is a probability of duplication that is happening.
But you don't have any selection.
There is no selection here.
Then how is it species dependent?
Exactly.
So then, because now you can alter these parameters, and based on altering those parameters, they actually try to infer. In the case of Drosophila, they have actually tried to show that. Let me get that here. So for instance, in this table, they have these 12 Drosophila species, and sure. Yeah. So again, similar to the kind of data that Rakesh was talking about here, they're speaking of a particular, two particular elements, this Ru and Ru A elements, for which they know the size of that. So that's the size of those two. So that's basically length I. So you know the entire length of the string in which additions, deletions, or these can happen. And then they look at the number of full-length elements that were seen in this context, and also the partial transposable elements that were seen, with lengths which were greater than either 200 base pairs. So again, if you talk about hexamers and there are more than say 35 hexamers, so you have already crossed 210 base pairs in that context. So then they look at how many of these base pairs, or how many of these simple sequence repeats, or what was the size distribution for many of these. So then this is the data that they have collected or was publicly available in this context, and then they try to match this data using that model. That's the general idea that they're trying to do here.
But what are the parameters in the model? There's the insertion rate, deletion rate, and those things?
Yes, insertion rate, deletion rate, and then you have F, which is the length of the gene, in which you'll actually see more deletions or more insertions in that context. And then you can also, again, one of the underlying assumptions is that deletions of each different size of base pair is equally likely. That is also, you can also have a weighted factor in there. Similarly, insertions of each different sequence, you can have a weighted factor there. So what I'm saying is that this model can potentially be generalized, and we can have more parameters built into the model with which we can try to fit some of the data that Rakesh was showing.
No, but won't that be a boring task? I mean, so
yes.
I mean, I think to address Rakesh's question about the snake and the and what the you would like to put some fitness.
Some fitness,
no?
Yes.
And then and that makes it more interesting.
Yes.
Problem.
So again, this is a very sort of first generation model which doesn't have any of those complexities, and they are somehow still able to, at least for these two genes in these species, match the results that were seen experimentally. But again, this is highly context dependent, highly species dependent, and the questions of fitness do definitely come in. So none of this selection has been specifically done. So again, they just talk about the distribution of these total transposable elements in these 12 species. So it's a starting point in that context, but then looking at maybe the same gene in different species and then going to questions of fitness is something which might be potentially interesting.
I find there are problems in this study. The reason is, I don't know what kind of transposons they're looking at.
Right.
So there are two types of transposons. One is DNA transposon, one is retrotransposon. So for DNA transposon, you don't have to rely on what is the rate of transcription. But for retrotransposon, it has to transcribe to RNA and then it becomes DNA.
Right.
So only retrotransposon works in the copy-paste mechanism. A DNA transposon works.
So they have a copy-paste mechanism.
Okay. So DNA transposon works in cut-paste mechanism.
Yes.
So you cannot have increase or decrease of length.
Right.
So you can only study these in the retrotransposons. Not
using this framework, yes. But this framework can be adapted to study the cut-paste mechanisms, where once you have so what here is basically insertion and deletion are decoupled. But in a cut-paste mechanism, once an insertion is happening, once a deletion is happening, then you can also change the frequency of that particular string and then add it to the other. So if you couple those, then you can study a cut-paste mechanism.
That's right, but when you're comparing across species,
yes,
the way you can have a deletion is if you have several such elements arranged in tandem, then you can have a homologous recombination, and then one repeat can be lost. So the rate of homologous recombination and rate of transcription of these elements can be a factor of, or can be quite species specific. So if you really want to compare such an event, the best thing to do is to do an experimental evolution type of experiment. You just take one species in the lab for many generations and then do sequencing and see what kind of changes you're seeing. The other problem I see in these kind of studies is if the regions are very long and repetitive, we find that there are a lot of assembly errors in these sequences. So unless you are absolutely sure that there is no assembly error, and such sequences, such organisms are very rare to find, I don't think these assumptions would lead it anywhere.
So the assembly error is an experimental measurement error. I mean that I'm not sure how mathematical modeling can sort of tackle that aspect. But going back to the other potential mechanisms which you were talking about, which can contribute to changing this distribution. So in fact, that is one of the advantages of doing mathematical models. It's basically what you were talking about is a set of experiments. These are just being done at a much quicker and much cheaper scale. So we can explore all these possibilities, have some weight factors associated with each of the different mechanisms and their interdependencies, and then perhaps try to rule out some cases that this mechanism might not help explain some of the observations that you have currently. So yeah, so it's supposed to narrow down the solution space, not necessarily arrive at a solution which is going to explain everything, because that's not the case.
So I'm still confused about that. Suppose you write some model and you get the data and you fit the model and you get some, what do I learn from that?
So you learn from that basically what is the probability of insertion, like how particularly this distribution of transposable elements arrived. Was it largely by insertion? Was it largely by deletion? Was it largely by duplication?
Yeah,
these are the only three mechanisms they talk about here.
Sure, but that depends on the complexity of the model. You can put this thing, that thing, and you can put a hazard kind of things. But then I'm still confused about, I don't see what you learn. I mean, this is one mechanism that you showed some microscopic model can reproduce that distribution.
Right.
But can you compare it against data? I mean, there's no time
There's no time element here. Yeah, so again, I mean, I'm not endorsing this model in any way. I'm just using it as a discussion point to talk about. But then, for instance, the data that Rakesh presented, where some particular transposable elements were much more enriched as compared to others. So then we can build a more detailed biochemistry incorporated into these models and then see if we can actually reproduce that. So some of those questions. Yes, right. And then
yeah.
Yes, right.
Does that come out?
Again, they have done only one species. Yeah. So they have not really tried all different, many different.
Generically, that was the case, right?
Yes.
Yeah.
I don't know whether it's a polymerase cleavage or some kind of intermediate, how we get this finite, are we clear about it?
Yeah, they don't really have that very clearly. They do some of these sort of on a log scale. They talk about these different length distributions that they have simulated in different cases and see which of them tries to match the experimental better. But yeah, so it's it's a starting point, but some of these aspects which can, again, as Rakesh was talking about, that using the biochemistry, we can perhaps guess the function of what particular that SSR might be, such as the enhancer blocker, because if there are two different genes which have these different functions, as we talked about the ILS and others, so similar sort of educated guess mechanisms or going into more detailed biochemistry can be potentially built into these models to then compare them.
That has this enhancer blocker activity. In fact, people have mapped even which part of the transposon is involved in this. So there are some 12 binding sites for a protein called suppressive hairy wing. It doesn't matter. So, and this 150, about 250 nucleotide, very strong blocker. Wherever this transposon lands, the reason for the mutation is now enhancer is not able to talk actually. So, yeah.
Yeah, so that was one of the questions which we were discussing, because one of the things which comes up continuously in SSR literature is that they are distributed in a non-random way. So as also Rakesh showed, that they were very low in exons and then higher in introns and intergenic regions. So one broad question is that why do you actually get this relative, completely not non-random process, but there's some distribution. And the other was the size distribution aspect. Why does it vary with respect to the species? What different factors are contributing to that? So for the second question, this is at least a starting point where we can start to formulate questions in this particular way and then build our way up.
Phenomenological studies, presumably there is some region of parameter space where they were able to match with the
Yes.
X. So suppose if you were to repeat and try to see if the same parameter space is also the valid one in a different species, then that will lead a lot more strength to these phenomenological models, right?
Yes. And
so has such studies been done, or?
Not that I know of. This is the, I mean, this group has published one or two earlier papers, but even simpler abstractions than this one. So, and again, going back to that point, perhaps the distance between the parameter regions in a multidimensional setup can in somehow be correlated to, or we would expect them to be in somehow correlated to the distance in terms of speciation, if there is some evolutionary component that is acting, or that can be one parameter contributing. So we can look at all of those correlations once we figure out this, because this, much of the data, and please Rakesh correct me if I'm wrong, much of the data they actually present here is perhaps available for different organisms in this case, for different transposable elements. So there is potential of generalizing this model and making it less phenomenological and more mechanism based, having some hybrid in there, and then figure out these parameters and see where different species for different genes lie in that parameter region, how similar they are. Is the intra-species differences more, or are the inter-gene differences more? At which level is selection really happening? Yes.
Is there a slide which talks about the comparison between the model results and the data?
Yeah, I'll get to that. Yes. This is the slide. This is the data that they have, where they basically aggregate the counts for all different 12 Drosophila species, and then they compare the distribution that was obtained from the data, which is this empirical distribution, to the exponential steady state analysis fit that they had found. Yeah, so the data of interest, so the blue is the data that they have gained, so that's the aggregate counts. So this is probability, this is some cumulative distribution, so the x-axis is the length of those different cases. Yeah, so the x-axis is the full length elements, and the y-axis is the probability of, just let me make sure. They had a description, just a minute. So yeah, so they both of the, so there are two figures. One is this one and then the next one. So here the x-axis is time in terms of generations, and so here there is no data. This is basically they are looking at different components here and then trying to see how those different curves look like. And in this particular plot, so yeah, so the x. So the x-axis is this length of the element that they're talking about, and the y-axis is the probability of finding a transposable element greater than that length.
model to the variable that Rakesh showed, which talked about number of, right?
Yes, if you're talking about hexamers and that graph went all the way to 20, 25 or so.
For the tetramer, right?
Yes, yes.
You had shown this figure. Non-monotonicity.
Yes, this doesn't. Yeah, exactly. So again, this is too simple a model to talk about. The experimental data, yes.
The data does not show any non-monotonicity.
Because they've looked at a particular transposable element. And this is cumulative.
No, you will still see a change in the slope. Right? Even a cumulative
Yes.
will show that there is non-monotonicity in the original.
Right.
So,
yes.
Just a comment. If there's p-value 10 to the minus 14, my eye tells me that the data on the field are not.
That's p-values have their own issues, as Vidya pointed out. So. But yeah, this is the only data that they actually talk about in detail. Yes. And then again, they have all these different guesses, but in terms of comparing different predictions, but in terms of comparing with the experimental data, this is all they have, where they have talked about all these 12 different Drosophila species in that case.
It seems to me that we want to get, this is the goal, get in a much simpler way.
Yeah.
Even a hand waving.
Yes. But yeah, going to the questions of non-monotonicity and then how does sort of species specific, because again in Rakesh's data, I think it was zebrafish which had a much longer tail, but the others sort of peaked around at a relatively conserved cases. So there have to be possibly some common factors which are acting in there and then some parameters which are diverging the solution space. So this was one quick paper I wanted to talk about. There was one another I'll just quickly flash in before we head for lunch in 10 minutes. So this is going to the other end, which is understanding these, yeah.
What is the question that you want to answer? I'm still confused.
So here the question, again, the two questions which, the previous one. Yeah, so the previous one was trying to, so the previous one was basically trying to see if with this simple model that they have, can they actually explain why or the distribution of those transposable elements in that relative to that particular gene in that particular species. That's it. That is the scope of the question. I agree. I'm not saying this is the best paper out there. That's it. Yes.
It's a methodology of thinking.
Yes.
What does it tell you?
So so that this again, based on what I understand on this paper and discussion with Rakesh, this tells is the relative contribution of these different molecular mechanisms which Kostov and Rakesh were talking about, which might be contributing to the generation of these SSRs. Because why are these SSRs present is not yet clear. I mean, they are present and they contribute to almost 20% of our non-coding DNA, and they have some functions. That is something which is sort of being taken forward, but how do they emerge? Why is there a specific pattern to it in terms of distribution across different species, both in terms of where they are and in terms of their sizes, is something which at least we thought is not really clearly understood and is a particular domain where modeling can perhaps be useful. Does that help? Okay. Yeah, so this is again taking it to the other extreme, which is understanding these simple sequence repeats well enough to be actually constructing synthetic circuits based on these simple sequence repeats and then controlling the behavior of this function. I'll just zoom this in. So here again, the goal was completely different. The goal was to, if we can actually construct synthetic circuits based on what we understand about these simple sequence repeats, and then develop these circuits or have this idea of targeted mutagenesis, which is, can we actually optimize the behavior of a given gene network via what they call a directed evolution process. So what they have done here is They have constructed multiple different expression libraries by actually constructing large number of combinations of these simple sequence repeats, inserted them in a particular genetic circuit, and then see that how many of them were actually able to give rise to the behavior which is generally expected from the circuit. And that the circuit that here they are looking at is again one of the most well-studied circuits, both sort of theoretically and experimentally, is this double negative feedback loop. So this is again seen in multiple developmental contexts where there is a self-fate decision that has to be made, a binary decision that has to be made. This was the first synthetic biology circuit which was constructed in the year 2000, where they actually showed that this system can give rise to bistability and cells can be in two states, which are sort of A high, B low, and the other one being A low, B high. So a particular given cell population is likely to exhibit some sort of a bimodal response in this particular case, some cells in this particular state and some cells in this particular case. Now, if you were to assess that experimentally and have the number of cells in the levels of A, so this would look something like this, where you can actually see two peaks in the levels of A or in the levels of B. So that is what they have actually been able to look at this. So what they did was they had these simple sequence repeats, and these simple sequence repeats, as again are known to have a high mutation rate and a strong bias for these insertion or deletion mutations, they were actually able to insert it in the, and I'll get to the figure there, in the ribosome binding region. Yes. I'll get to that in a minute. Yeah. Yes. Yeah. Yes. So they basically, what they have done here is since these simple sequence repeats can be of different length, it as Rakesh talked about anywhere from monomer to hexamer, and they can have different sequence of nucleotides in which they are attached. So what they do here is that they actually synthetically generate those different ones. So let's say there can be something, this is one example, you can have a large number of combinations here. So they actually generate these simple sequence repeats which can vary from anywhere from 1 to 6. And then similarly, you can have different combinations which are possible here. Then they actually insert that particular setup, the simple sequence repeats, in a particular given circuit, this particular initial circuit. So just before the ribosome binding site, just as Rakesh was talking about, just between the enhancer and the promoter, this particular simple sequence repeat was inserted, which actually affected the function here. So similarly, they have inserted this different combinations that they have generated in here and then seen the effect of that on the behavior of the circuit. That's the general thing. So what they mean, yes. So what they mean by expression library is just all different combinations that you can get here. That's all. Yes. Right. Yes. 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 So their final readout is, since they're looking at this bistable circuit, their readout is bimodality. That is the readout. So each of them is then associated with some fluorescent marker, and that fluorescent marker, the distribution of flow cytometry of that is what is they're looking at. So if you were not to do this experiment, this is an experimental paper. This is not simulation. This is synthetic biology. So taking it to the next level. Sorry, I should have made it even more clear.
So in that case, if that is the case, then they have identified one particular circuit in E. coli, which is of that type.
Right.
There are two genes corresponding to A and B,
yes,
which are mutually repressing each other.
Right.
And their regulatory regions have these various repeats.
Exactly.
Right?
Yes.
And if I understand correctly, they are making the mutation in the regulatory region where these repeats are, and trying to see whether you can get that bistability pattern become better. Right?
So they are synthetically inserting these ones. Just as Rakesh synthetically inserted a particular DNA sequence in between the enhancer and the promoter region and actually showed that this particular simple sequence repeat was behaving as an enhancer blocker because the gene was no longer transcribed. That is very similar to what they are doing.
So they're modifying the genome.
They are modifying the genome, yes. Yes.
And the effect of that on the readout.
Exactly. Yes.
And they are claiming that by suitable insertion deletions, you can get a better.
You can alter this curve in different ways. Yes, that's what they're saying. Yes. I'll get to that. Yes, right. So right, so that's where they embed these. So actually, they have done only mono and dinucleotides, not the all 6 ones. And then they also look at whether these particular sequences were stable. Were they stably inherited by generations? Again, going back to Kostov's point, that for how many generations can you actually see the same simple sequence repeat in there? So here again, the goal is to see if these simple sequence repeats can be used to change the dynamics of a given circuit, the dynamics of a circuit which we relatively well understand as compared to others. Yeah. Yes. So for E. coli, this happens on a scale of hours. The next cell division, and then this generation. For mammalian cells, it's of course days. So that's why they were able to trace this over multiple generations. Yes. So these are the circuits that they have considered. So these are the different spacer motifs that they have considered. In this case, only inserting A's multiple number of times, only T's, A and T, and then that continue, A and C, and then that continues. Those are the only combinations they have considered in this particular case. So once you can, again, as Apratim was talking about, once you can insert this and have some fluorescent protein being driven by that, you can actually take that as a readout of the cell. So here, the two aspects, the two proteins they are finally measuring are being measured. One of them is being measured by RFP, so red, and the other is GFP, which is green. And then they look at the distribution shown later. So they have made these changes, the simple sequence repeats. They've inserted these simple sequence repeats in this particular circuit, which is being shown by the P-lac-I, P-tatar aspect here. So this tatar and lac-I, which is, they're inhibiting each other. And then their expression is driven by GFP and RFP in this case. So what you can see in the graph on the B panel is just a representative case where you look at how many cells are expressing RFP and how many cells are expressing GFP. So again, this is closer to 50%. So there are almost equal number of cells which are expressing those. So the two stable states of the system that we are talking about have relatively similar stability. And then what they do later is they actually vary how many number of repeats of T were present in gene 1 and how many repeats of A were present in gene 2. So then you can do this sort of similar to a titration experiment and vary the relative stability of the states. So again, you can go the system all the way where the cells are all green and drive the system again to a state where cells are all red. So you can change the relative stability of the states by inserting this particular case. So you're changing how much transcription is happening, and this again goes back to what Rakesh was saying, that there is a function of that, and here they have shown a way where you can actually modulate that function synthetically, so we understand that well enough to do that. This is just another example where they varied the number of sequences here and then here. These are just different libraries that they're talking about. This is only on this kind of circuit. Yes, again, but you can actually build this in
Interesting.
Yes, because yes, now you can actually fine tune, because otherwise the ways to fine tune are to change the binding coefficient or the binding strengths between the promoter and DNA of these ones. What they have done here is that by inserting those simple sequence repeats, you can do something which perhaps doing in a more molecular biology dominated way might be much more challenging and involved. So the other way they have tried to come up, or they see, is you can actually insert these or delete these. They have only done insertions, but perhaps this can also be generalized for doing deletions and amplifications and see how you change that. Sorry?
The kind of experiments you can do.
These are kind of experiments which can be done, yes. Yes, because again, this again, what Rakesh was talking about, very actually insert and see how the difference of, this has been done in E. coli, but Rakesh has done this in Drosophila also, so this can perhaps be looked at even complex organisms. They have some control experiment that they did, but to answer your question, I think it is specific to these repeats. They have some, yeah.
So do these repeats?
When you say Tn,
yes,
right? I mean, if you can go, T sub n.
Yes.
That n stands for the number of T's.
Yes, that were inserted.
And these T's are inserted contiguously to each other.
Yes.
Mm-hmm. And the location in which these T's and these A's are inserted, the starting location where they're inserted, that has been fixed in advance?
Yes, that again goes, so that has been fixed in advance, yes. So this is where they're inserting the space, what they call spacer. It's basically a different simple sequence repeat. So they are inserting this just before ATG. They do it all the way up to 20, and they only try again a limited number of combinations, but what they observe is that this is a way to control the transcription of those genes. So again, going back to the functional aspect, what is the function of these SSRs? So here they insert it in a titrated way, and this, you can actually see the effect. This can be quantified and then plotted and see what kind of curve do we get.
I see. So, if I understand, I think you probably mentioned it, but just for clarification, what this shows is that you don't have to do any changes in the transcription in the A's and B's themselves.
Exactly. Yes.
You just have to change the number of repeats which are appearing in the promoter region,
right.
And by suitably changing that, you can span the whole range of behaviors from monostable A to bistable to monostable B, right?
Yes.
Mm-hmm. So it demonstrates the flexibility and power of modifying the regulatory consequence from these repeats.
Right.
That's right.
So this directly speaks to the functional implications of those repeats. I mean, we might still need to have a long way to go from sequence to function in the context of these repeats, but at least this is a relatively large-scale experiment as compared to what Rakesh was talking about, that one single enhancer blocker. This is a relatively large, and this can be done in a more titrated way, at least some semi-quantitative way, and then you can fit those. Yes. Yes.
They sort of present similar scenarios.
Yes.
Have those phenomena been mathematically modeled? I'm not aware of it, but some of you may.
Yes, so the lac operon, the lytic lysogenic aspects have been mathematically.
No, lac operon is different. I'm talking about lambda switching. There's a lot of cooperativity sequences, repeats of the nature you are indicating. And yeast mating type switches are a different scenario. Maybe Kaustubh can reflect on this to some extent.
The first one I definitely know has been modeled extensively, both deterministic and stochastic, and there have been multiple formulations of that model as we have come to know more and more of molecular biology underlying that. I am not aware of the second one.
Yeah, I don't know